Hello students, we are back. This time to talk about not mitosis with a T, but meiosis with an O. i.e. 2 divisions, 4 cells made, only used for reproductive cells. Let's get to it.
So this beautiful little foldable is what I ideally wanted us to go over in meiosis. However, we've had snowpocalypse. We've had hybrid learning. We'll try to do our best with what we get. As you can see, meiosis takes 2 divisions. Meiosis One, where I still go through prophase, metaphase, anaphase, telophase. And then I take those 2 cells that I make and I go through meiosis Two, where I go once again through prophase, metaphase, anaphase, telophase. And that's how you end up with 4 haploid cells. If you already had this handout in class, then you're going to want to take the handout, skip ahead a little bit further in the video till you get to here-ish. That way you can figure out how to fill out the rest of it. If you didn't get the handout in class, once again, I'll show you guys how to make things using a blank sheet of paper. I'm trying to figure out a good way to digitize this one. If I do find that out or figure it out, I will send out a reminder to the cohort, letting you know that there's a digital version available on the hub as well. I think what I might do actually is scan this page and put it on the hub for those of you who can print it out. But for those of us who are less fortunate, do not have a printer at home, let's take a blank piece of paper. Let's fold it in half hot dog style. And what we're going to do is, if you want to, I would say go ahead and draw yourself a little line across that fold, dividing your paper in half. You're going to do meiosis 1 on top and meiosis 2 on the bottom. Meiosis starts with a cell. Meiosis is only for sex cells or reproductive cells, and the originators for those are going to be called germ cells. Yes, like germs or bacteria. This germ cell is going to be happily an interphase, i.e. G1SG2. While chilling there in interphase, he is going to have all of his DNA nicely contained within a nucleus. At this point in time, the chromosomes are pretty loose and uncondensed, so they're all just kind of chilling there, a little bit like a big mess of spaghetti. My cell is also going to be considered diploid, as in he's got 2 copies of every chromosome. Or homologous pairs. Alright, let's move on. After interphase, I've gone through G1, I've gone through S, I've gone through G2. I am going to move into our first stage of meiosis. Just like the first stage of mitosis, this is going to have the beautiful name prophase. But this is not just prophase, this is prophase 1, because there's 2 divisions in meiosis. So I need to have, I have a prophase 1 in the first division, prophase 2 in the second division. Just like prophase for mitosis, the nuclear membrane will be breaking down. In prophase 1, I still have sister chromatids paired together. So we can talk about having a larger pair and a smaller pair. But they do something slightly different here in comparison to mitosis. They will find what's called their homologous sister chromosomes, or chromatids. Yeah? And they will pair up with their homologs as well. Okay. So, what is a homologous chromosome? Why is this so important? Homologous chromosomes are very similar in nature. These guys are going to have basically the same size. They're going to have genes that code for the same trait. much you're going to have one from your dad and one from your mom, which means that though the genes code for the same trait, they might be slightly different versions of the genes. Meaning that while these chromosomes are very similar to each other, they have slight differences. So they're not identical like the sister chromatids are. Instead, they're homologous. They are close, but not quite. Okay? When the homologous chromosomes do this, they are going to be forming what's called a tetrad. Tetra often stands for 4. So a tetrad is going to be when you have a linking up of 4 sister chromatids. Okay. One last thing happens here in prophase 1 that's really important, and that's the fact that these guys are so close together, they're going to do something called crossing over. And that is when the sister chromatid from one homologous chromosome is literally going to cross over with the sister chromatid from another homologous chromosome. So it ends up looking something kind of like that. Okay, that was a lot for prophase 1 of meiosis 1. However, don't worry, because after this, everything just follows the prophase, metaphase, anaphase, telophase, prophase, metaphase, anaphase, telophase, very simply and straightforwardly. It's really completely about the setup in prophase 1 that makes meiosis so different from mitosis. So let's take a quick pause to compare. In mitosis, I'm going to title both of these so you guys know which one I'm talking about. This is mitosis. Over here we got meiosis 1. Okay. In mitosis, I see the spindle fibers attaching into my sister chromatids, but they're not pairing up with their homologs. Instead, their homologs are going to be somewhere down here. Another homolog will be up here. We're not seeing this tetrad formation where you get your homologs paired together. Also, I don't see in mitosis any crossing over or any sharing of my genetic information between my homologous chromosomes. And that's simply because they're not arranged in their tetrad formation. Okay. So here's where I'm going to get genetic variation, and this is also the reason why I'm going to get haploid cells with half the amount of DNA. Let's roll forward, and you guys will see how this plays out. After prophase, PMAT, I go to metaphase 1. And in metaphase 1, very much like in metaphase of mitosis, I'm going to line up in the middle of my cell. Except this time, I am lining up tetrads of homologous chromosomes and not just chromosomes themselves. It'll look something like this. You'll still have your centrioles. The centrioles will be attached, but this time instead of attaching to the sister chromatids, they attach to homologous chromosomes. So let's pretend we've got one pair here, and then we've got the sister with the crossing over going on over here. Okay. Lining up my tetrads this time instead of just individual chromosomes. Told you we were going to go faster. After metaphase, what happens next? These guys are going to get pulled apart, right? In anaphase. Anaphase 1. Once again, I've got my centrioles. They're attached to the homologous chromosome sets, this time with their spindle fibers. On one side, I have one set of homologous sister chromatids. On the other side, I have another set of homologous sister chromatids. over here, because of crossing over, I've got mostly this guy, but he's stolen a little leg off of his homolog. And the same thing is kind of happening over here. Mostly okay, but he's stolen a little leg off of his sister homolog. So now you can kind of see where we're going to get some genetic variation or mix-up happening. We're going to say this is homologous chromosomes. Homologous. And this is going to be homologous chromosomes are separated. Okay. P prophase, M metaphase, A anaphase, P mat. Our last stage is going to be telophase. In telophase, we can see our cell starting to maybe widen out a little bit as I basically start to reform my two new nuclei. And pay attention because in each of these two new nuclei, we have not individual chromosomes, but kind of sets of sister chromatids. I'm going to draw it this way first. But don't forget that you still have a little bit of the sister chromatid shared over by crossing over. Last but not least, after telophase 1, we've got to go through cytokinesis 1. I'm going to have my cleavage furrow pinch my cell together, and I'll form my two daughter cells for meiosis 1. Something along the lines of this. I apologize, I misspoke earlier. The nuclear envelope doesn't actually reform because you're about to go through your division again, so it wouldn't make sense to form it and then break it down right away. This is what makes telophase 1 a little special. Okay. We're halfway there. What have we done so far? We have gone from prophase 1, metaphase 1, anaphase 1, telophase 1 to cytokinesis 1, and we've got two daughter cells. These two are considered haploid already. Because I've separated my homologous chromosome pairs. However, they've got an extra chromatid duplication, so we're going to have to basically take them through mitosis one more time to make them truly haploid. All this time I'm talking about my homologous chromosome, separating that green from that blue, or the mom's chromosome from the dad's chromosome. Now when I start talking about meiosis 2, I'm going to be talking about separating sister chromatids. So one chromosome here from the other chromosome here, the ones that are usually the replication chromosomes made from DNA replication. Let's do it. Let's go to meiosis 2. In meiosis 2, I'm going to be talking about these two daughter cells. So at this point, I'm actually going to cut this apart for myself, tape it on here, and continue the story. Meiosis 1 has happened. Meiosis 2 shall now happen. Let me go find some tape. Let us continue the story. So, with my two haploid daughter cells, I'm going to basically go through mitosis a second time, and I'm going to call it meiosis 2. First stage
is always prophase. This time though it's prophase 2. I'm going to be dealing with both daughter cells at the same time. I'm going to watch the spindle fibers attach to the sister chromatids and the centromeres just like they did for mitosis. Remember, it's prophase, so they're not quite aligned in the middle of the cell yet. They kind of still get in there, still attaching in. Don't forget that we've got these funky little guys here that are carrying bits of each other from prophase 1 and crossing over. After prophase, we always have metaphase P mat. This time, since I'm in the second division, I call it Metaphase 2. In Metaphase 2, still got my 2 daughter cells. This time, I'm going to align my sister chromatids. What does that look like? Let's give ourselves a quick sketch. You get the idea. I'm going to do one and then I'm going to skip forward. Just try to make this video a little bit shorter. So, just like in mitosis, I'm going to kind of see my 2 chromosomes connecting on in. Don't forget, he's a recombinant. He has this little thing there. Same thing's going to happen for this guy. I'm going to skip forward. After metaphase 2, we're going to have P mat anaphase, the phase where things are pulled apart. It's meiosis 2, so it's anaphase 2. Can maybe draw my cells getting a little bit more elongated as things get pulled apart. Centrioles and spindle fibers, always at the ready, doing the actual work, pulling these things apart. And this time, just like in mitosis, I'm going to separate my sister chromatids. Ba-ching, ba-ching. It's going to look something like this. So I have one chromatid here, one chromatid here. I'm going to have one normal chromatid here, but then I'm going to have this funky guy who got a bit of his homologous chromosome from his tetrad at the beginning. Same thing is going to happen on the other side. I'm going to split apart my sister chromatids. And I'm going to have the funky guy over here, actually, in this case. The little bit of extra. A little different. These guys here are called recombinant chromosomes because they are basically homologous chromosomes that have recombined. And this is how you get that genetic variation in your daughter cells of meiosis, because you're recombining chromosomes there. All right. We're almost there, guys. Prophase, metaphase, anaphase, telophase. I think you guys can figure out where this is going from the rest of here, so I am going to kind of speed up my video. I'll do a quick time lapse of this. Alrighty, telophase. Got 2 elongated cells. I'm going to form 2 new nuclei inside of these using my newly separated sister chromatids and recombinant chromosomes. Little cleavage furrow will form. Oops, forgot the description for anaphase. Remember, sister chromatids are separating. Alright, cleavage furrow forms, and we have cytokinesis, where we actually split the cells apart, forming 4 daughter cells, each with slightly different combinations of chromosomes. Phew, finally reached the end. At the end, I have 4 1, 2, 3, 4. Haploid, because they have half the amount of DNA they had at the beginning. And I can test that because I had like 1, 2 chromosomes here. All the way at the beginning, I had 1, 2, 3, 4 chromosomes. So 1, 2, 3, 4 chromosomes becomes only 1, 2 at the end. So 4 haploid. Now, if I look at each of these cells, I'll notice that they have different DNA. This has one homologous chromosome set, another homologous chromosome set. This one has recombinant plus moms, let's say, and then a recombinant plus dads. They're not the same, so we have 4 haploid non-identical. Gametes. What were gametes again? Eggs and sperm. Remember, this process only happens for making egg and sperm cells. It only happens when you are trying to reproduce. Now you have your super long guy. You can glue him into your notebook and then kind of fold him over so that he actually fits in. But I think it's nice to have the overly sized long foldable so that you can remember that meiosis is quite a bit longer than mitosis. Yeah? For these inside flaps, I just kind of put down some things I wanted to remember about meiosis. If you would like to include that and also what homologous chromosomes are, you can. If you want to talk a little bit about like that cool crossing over that was happening in prophase, you can. If you also feel like you understand the process pretty well just from your flow chart and what we've covered in class already, go with that. Either way, I expect you guys to have at least a basic understanding of what prophase, metaphase, and anaphase and telophase look like for both mitosis and meiosis, and kind of the basic differences between the two in terms of the processes and in terms of what they're used for. I'm going to post full notes like in picture format of these also on the hub. Please reach out if you have questions, rewatch these videos as many times as you need to, and I will see you guys on Friday for the final wrap up of mitosis and meiosis. Good luck!